Hi everyone, today a video on biological chemistry, specifically looking at amino acids, peptides and proteins. So amino acids are the building blocks of proteins. Proteins are polymers and amino acids are the monomers that we make these proteins out of, these polymers out of. And proteins are themselves a vital part of all living systems. So amino acids have two functional groups on them. They have a carboxylic acid at one end and a primary amine, that's an amine with one carbon group attached, on the other end. And there are 20 naturally occurring amino acids, and they are all what we call alpha amino acids. And what that means simply is that the amine group is attached to the carbon that is next to the carboxylic acid. Essentially, we have our carboxylic acid carbon, another carbon, and that's the one that our amine is attached to. And what that means is that we can write a very general formula for an amino acid. We have an NH2 group at one end, a carboxylic acid, a COOH group at the other end, and then in the middle we have a carbon atom which has a hydrogen connected, and then simply we can write R. And it's this R group that is different for every of those 20 amino acids. They all have that same structure. Now, as you can see, that central carbon there has four different groups attached to it: our amine, our carboxylic acid, a hydrogen, and whatever our R group is. So what that means is that the molecule is chiral. So actually, for 19 out of those 20 carboxylic acids, they are chiral molecules. The only exception is glycine, where there are two hydrogen atoms on that central carbon atom, and therefore there's not four different groups. And so you can see on the right here, all of the 20 amino acids drawn out. And they can be classified into different groups. They can be non-polar, such as glycine, alanine. They can be polar, such as serine. They can have positive charges on them. They can have positively charged groups like NH3 groups, such as lysine. Or they can have negatively charged groups, such as aspartic acid. And so amino acids can be referred to by their systematic name, for example, glycine. They can be referred to by a non-systematic name. They can be referred to by a three-letter abbreviation, which for glycine is GLY, or simply by a one-letter code, G. So, as we said, amino acids have two functional groups on them. They have their amine group. An amine group is a basic group. It has a lone pair of electrons that can accept protons. And we have our carboxylic acid group, which clues in the name, that's an acidic group. It has a tendency to lose a proton. And so if we have a molecule where we have both a basic group and an acidic group in the same molecule, we can kind of get an acid-base reaction going on within that molecule. The carboxylic acid group losing a proton and it being picked up by the amine group in the same molecule. And when this happens, we say that this is something called a Zwitter ion. And amino acids exist as Zwitter ions. These ions have a permanent positive and a permanent negative charge, but overall it's a neutral compound. And so amino acids are ionic, so they have high melting points. They dissolve well in polar solvents such as water. And so a typical amino acid is just a white solid at room temperature, and it behaves like the kind of general ionic salts, generic ones like sodium chloride. We can also think about how amino acids are going to react in acidic or basic conditions. If we're in strongly acidic conditions, for example, if we're in a solution of acid and we put our amino acid in, the amine group will pick up protons using its lone pair. It will act as the base, and so it will end up being protonated. And that's what we see on the left-hand side of this reaction scheme at the bottom. We've protonated that NH3 group. We have a positive charge, an actual cation. In strongly basic conditions, well now, if we put it into a solution of base, for example, it's the carboxylic acid part of it that reacts. It will act as a base, it will lose a proton and become deprotonated. And that's what we see on the right-hand side here. We've actually made an anion. We've deprotonated that group. And then generally at kind of fairly neutral pH, the Zwitter ion is what exists. We've lost the proton from the carboxylic acid, but it's been picked up by the amine group, the same molecule. So it's not a cation, it's not an anion, it's a Zwitter ion. Permanent positive and permanent negative charge, but overall neutral. So, as we said that amino acids eventually end up forming proteins, well, the sort of first step in that is that amino acids will link together to form peptides. Now, polypeptides are molecules that contain up to 50 amino acids, When there are more than 50, they are called proteins. So really, there's no real distinction between a peptide and a protein, just that a peptide is generally smaller. But the chemistry of them is the same. And so the amine group of one amino acid can react with the carboxylic acid group of another. Essentially, it's a form of an acylation reaction that we might have seen before. And this forms what we call an amide bond or a peptide bond. So it's a simple nucleophilic addition-elimination mechanism. And we end up forming an amide or a peptide bond. And so compounds that are formed by the linkage of amino acids are called peptides. Now, if we have a peptide that has been made from 2 amino acids, we call it a dipeptide. So it's slightly sort of funny naming that the molecule is called a peptide. If it's made from 2 amino acids, it's called a dipeptide, even though it's just one molecule. The di bit stands for how many amino acids made it. If it was made out of 3 amino acids, we'd call it a tripeptide, and so on. Now, importantly, if we've made this dipeptide from our 2 amino acids, remember that there's 2 functional groups on every amino acid. So while we've used up the carboxylic acid end of one and the amine group of the other, once we've made this peptide, there's still a free amine end and a free carboxylic acid end. And so this can continue to react to produce polypeptides. Now, a particular protein will have a fixed structure of amino acids in its chain. This is what we call the primary structure. It's simply the sequence of amino acids that make it up. And so polypeptides and proteins are what we call condensation polymers. It's where we have eliminated a small molecule, in this case water. When our carboxylic acid and our amine group react together, we lose a molecule of water, i.e. a condensation, and we end up forming our polymer as the link forms. Now, we can also go the other way from this. So if we take our protein or our peptide and we boil it with a concentrated solution of hydrochloric acid, for about 24 hours, it will break down into the mixture of the amino acids that made it up, the constituent amino acids. And this is called a hydrolysis reaction. We're using the water to break open that peptide bond. So this means that proteins are biodegradable. And this is one of the big differences here between condensation polymers and through addition polymers that we've seen with alkenes, that the polymers we make from alkenes through the addition polymerization are very long chain saturated alkanes. They're not biodegradable. Whereas these ones that we made through condensation polymerization, these peptides and proteins, because they have this polar sort of linkage in the middle, it means they're biodegradable. They can be attacked by water. So looking at the structure of proteins, proteins have pretty complex shapes. And these shapes are held in position by non-covalent forces. So not actual fully covalent bonds, but non-covalent forces. And these are the kind of intermolecular forces that you've seen before. So hydrogen bonding, sulfur-sulfur bonds, for example. And the shape is vital for a protein's function. If the shape changes, quite often it loses the ability to carry out whatever task it needs to do. It loses the ability to be able to function properly. Now, many proteins are helical in shape. They have a helix structure or spiral. And actually what holds this spiral together, this helical shape together, is hydrogen bonding along the chain. So on the example at the right here, we can see that we've kind of coiled this around, and where one NH group is kind of pretty well positioned to be able to hydrogen bond to a CO, a carbonyl group, somewhere further down the chain. And so if we were drawing this out properly, we'd make sure that we show our delta positive on the hydrogen, delta negative on the nitrogen, delta negative on the oxygen, delta positive on the carbon, and make sure that we show our lone pair on our oxygen and then our dashed line to indicate the hydrogen bond. Now, another arrangement is called pleating. You might have heard of a beta pleater sheet. And this is also held together by hydrogen bonding. This time, though, the hydrogen bonding occurs actually kind of across the sheet as opposed to down the chain. And so the amino acids in a protein chain can bond together in a number of ways. We can have hydrogen bonding between the CO and the NH groups. We can also have ionic attractions between groups on the side chains of the amino acids. So this is where we looked at our amino acids at the start and we saw that some of them are positively charged, some of them are negatively charged. We can have simple charge attraction, almost ionic bonds almost forming, and also sulfur-sulfur bonds. So this is where there are 2 cysteine residues. So this is where we have an SH unit. These can be oxidized. They lose the hydrogen to form a sulfur-sulfur linkage, and this creates what we call a disulfide bridge. And so these are these interactions that can hold these protein chains together. So we talked about the primary structure being simply the sequence of amino acids along the protein chain, and we can represent it by the sequence of the 3-letter codes or the 1-letter codes for the amino acids. Now this primary structure is held together by strong covalent bonds. It's the actual peptide links that hold it together. So the primary structure is very, very stable, and that's why it requires the quite harsh hydrolysis conditions of boiling it with HCl for quite a long time to break it down. However, we can then move from this primary structure and actually start to form the shape of the proteins. So the next thing up is called the secondary structure. And this is where essentially we either form the alpha helix or the beta sheet that we saw on the previous slide. And so this secondary structure is held together by the hydrogen bonding mainly between the CO and the NH backbone groups. And since the hydrogen bonds are much, much weaker than covalent bonds, the covalent bond is an actual chemical bond, whereas the hydrogen bond is an intermolecular force essentially. This level of structure can be easily disrupted just by gentle heating or even really small changes of pH. And then finally we have our tertiary structure. So this is now where our alpha helices or our beta sheets can fold even more and start to interact together into a 3D shape. And so this tertiary structure is held in place again by hydrogen bonding. It can be ionic interactions, it could be sulfur-sulfur bonds, as well of course as van der Waals forces which exist between all molecules. So the key thing is that the primary structure is simply the sequence of amino acids that are in our protein, held together by actual strong covalent bonds. Secondary structure is when this chain first of all folds into alpha helices or beta sheets. That's held together mostly by hydrogen bonding. And then finally we get to our tertiary protein structure, which is our 3-dimensional shape, where our alpha helices and our beta sheets have started to kind of fold again and interact with each other because of either hydrogen bonding again, sulfur-sulfur bonds, or ionic interactions. There's also something called the quaternary protein structure, and this is simply where we have essentially 2 tertiary structures coming together from different chains interacting with each other. So 2 different amino acid chains that have gone through this, combining together. This is our quaternary protein structure. So one thing to bring in here to talk about how we can separate and identify the constituent amino acids is to use something called thin layer chromatography, TLC. And so it's very similar to sort of very basic paper chromatography, but the paper is replaced by chromatography plates that consist of a thin plastic sheet that's coated with a thin layer of silica or silicon dioxide, SiO2. And so this white powder, this silica that is on the plastic sheet, is our stationary phase. So what we do, similar to how we would normally do a paper chromatography, we have our mixture of amino acids. Let's say we had a protein chain and we've hydrolyzed this for 24 hours in boiling HCl. We've got a mixture of amino acids, but we want to know what amino acids are present. We take our mixture and we take a small spot and we place it on a line that we've drawn in pencil just above where the solvent line is in the tank. And so this is our starting line. The key thing is we need to put it in pencil because we don't want the ink from pens to start to split and interact with our results. And also the line must be above the level of the solvent to start with. And so the solvent is called the mobile phase. We then place a lid on the top of this beaker or tank to ensure that everything is sort of saturated with the solvent vapours, it can't evaporate off. And slowly the solvent will rise up the plate. And as it does this, it will carry the amino acids with it. Now each amino acid will lag behind the solvent front to an extent that depends on its affinity for the stationary phase. How much does it like that silica stationary phase compared to how much it likes being in the solvent phase? And so this ultimately depends on the intermolecular forces between the amino acid and the solvent. So the stronger the intermolecular forces are between the solvent and the amino acid, well, the further it's going to move up the plate. The more affinity it has for being with the solvent, the less affinity it has for being with the stationary phase. If the intermolecular forces between the stationary phase and the amino acid are much stronger, well then it will hardly move up the plate at all. And so when the solvent has almost reached the top of that plate, we're going to remove the plate, we're going to mark where the solvent front ended, again in pencil, And then we can visualize where the position of the amino acids are by spraying the plate with a developing agent. Essentially what this does is you spray it with something called nenohydrin, and this will essentially show the spots where the amino acids are in a color, or you can shine UV light onto the plate, and typically the spots can be seen there. Then what we do is we calculate something called a retention factor, an RF value. And we can calculate this for every spot. So RF is defined as how far the spot has moved from the baseline divided by how far the solvent front moved from the baseline. So you need to get a ruler out, measure how far the spot is from the baseline that you drew, measure how far the solvent front has moved up from the baseline, and then do one divided by the other. Now this is unitless. Typically we'll measure this in centimeters. So if we do centimeters divided by centimeters, we have a dimensionless value, and it will always be less than 1 because obviously the solvent front will have moved more than the spots will have done. And so the amino acids can then be identified by comparing these RF values to values obtained by pure amino acid samples run in the same solvent mixture. So these RF values can be found online or in textbooks, and you just simply compare the two, and that can tell you what amino acids you have in your mixture. Now it's not 100% efficient, because if you've got two amino acids that have very similar RF values, it could be that you only see one spot on your plate, when actually really there's two spots there. They've just overlapped and it looks like one spot. But generally, for a kind of first approximation thing, it's a very, very useful technique to be able to separate out and identify our amino acids. So, time to have a go at some questions. If you'd like to have a go, pause the video and the answers will be on the next slide. So we've got some questions here to give the names of some things, draw the structure of some bitter ions, draw some products when we have some reactions going on, and then talking about the kind of shapes of proteins as well. So have a go. So the first question then was to give the systematic name for alanine. So I've drawn alanine out. We have our amino group at one end, our carboxylic acid group at the other, and then on our central carbon we have our hydrogen, and the R group in this case is a methyl group, a CH3. Now when we name amino acids, it's the carboxylic acid end of it that takes the priority in naming. Carboxylic acids normally end up taking the biggest priority. So that's the end of our name. When we name carboxylic acids, we take the length of the chain, which in this case is 3 carbons long, so it's based off a propyl chain. Our suffix is oic acid, so we have propanoic acid at the end of our name. We then need to also make sure that we name the amine group on this. Now normally amines are named with the suffix amine at the end. However, because the carboxylic acid takes priority at the end, we have to use the prefix amino to talk about where the amine group is. And as we do with naming, remember everything goes in alphabetical order, not numerical order, and we have to number where our substituents are. So this is the amino group is on the second carbon, with the carboxylic acid being the first carbon, and so this is 2-amino-propanoic acid. We then have to draw the structure of the zwitterion of alanine, and so simply all we're going to do is we're going to deprotonate the carboxylic acid group, protonate the amine group, and so the structure is shown there. Then we're asked to draw the product when aniline reacts with certain species. So the first one was aniline reacting with methanol and concentrated H2SO4. Now this reaction is a nucleophilic addition-elimination reaction essentially. And so what we have here is our methanol acting as a nucleophile. We have our electrophilic part of that amino acid is the carboxylic acid group. And so we have a nucleophilic addition elimination reaction. Essentially it's an esterification reaction, and we end up producing the molecule shown there. Next was it reacting with sodium carbonate. A sodium carbonate is an example of a base. So if we've got a base, we need our amino acid to be acting as an acid, and so the acid part of it is the carboxylic acid end, and so that bit gets deprotonated, and then the structure is shown there. And then finally for C part 3, it was to react it with chloromethane in a 1:1 mole ratio. Well, now chloromethane is our electrophile, so we need our nucleophilic part of the molecule, and our nucleophilic part of our amino acid is the NH2, the amine group. And so this just undergoes a simple nucleophilic substitution reaction, and the structure is shown there. Part D was to draw the structure of the species present at pH 2. So pH 2, remember, the lower the pH, the more acidic we are. So this is a very acidic solution. So under acidic solution, any basic parts of our amino acid will be protonated. Now in this case, we've only got the one amino group, and so that becomes protonated to an NH3 group. If we had an amino acid that had multiple basic sites on, they would all be protonated. It's not just that amino group. Any basic site would be protonated. Part E was to draw the structure of a tripeptide of alanine. Now remember, a tripeptide means that we've made it out of 3 constituent amino acids. So we need to sort of bond together 3 alanines. So essentially we need to just put the 3 alanines together, and remember that they will come together by forming an amide or a peptide link. That's C double bond O NH linkage. And so the structure is shown in E. Essentially 3 of them together, and so we've ended up making 2 amide or peptide links. Plus we still have a free amine end and a free carboxylic acid end. Part F was asking us to what is the interaction that holds the shape of a helix together. And the main interaction is the hydrogen bonds along the helix between the CO groups and the NH groups. And then finally, question 2 was to draw the product of the hydrolysis of the tripeptide that was shown on the previous slide. And so the simplest thing to do is to identify where the amide or the peptide links are, where the CO-NH links are, and essentially to just split those in half. And remember that if we're hydrolyzing it, we're adding water. So we add OH onto our carboxylic acid bit, we add H onto our amine bit. It's the reverse of this condensation polymerization. And so we end up then forming these 3 amino acids because we had 2 peptide links. It's a tripeptide. So we're going to get 3 constituent amino acids, making sure that we have a free amine end and a free carboxylic acid end on either of them. So thank you for watching this video. I hope you found it helpful. There's lots more videos like this on my YouTube channel. If you found this video helpful, please leave a review on one of the tuition websites. I offer online tuition for chemistry, so please check my tuition pages for prices and how to contact me. And if you need any more help with your chemistry, you can always feel free to drop me an email. I'm always happy to help.